IDO1. IDO1 stands for indoleamine 2,3-dioxygenase 1. Gamma interferon has an anti-proliferative effect on many tumor cells and inhibits intracellular pathogens such as toxoplasma and chlamydia, at least partly because of the induction of indoleamine 2,3-dioxygenase. This enzyme catalyzes the degradation of the essential amino acid L-tryptophan to informal kynurenine. IDO activity in tumor cells may act to impair antitumor responses. Human tumors constitutively expresses IDO. The catalytic activity for IDO1 is D-tryptophan plus O2, which yields informal D-kynurenine and L-tryptophan plus O2, which yields informal L-kynurenine. The cofactor is heme. The enzyme regulation activity for IDO1 is inhibited by and MTHFR, also known as methylthiohydrantoin D-tryptophan, modestly inhibited by L1MT, also known as 1-methyl L-tryptophan. The catalytic efficiency for L-tryptophan is 150 times higher than for D-tryptophan. The pathways for IDO1 are amino acid degradation, L-tryptophan degradation via kynurenine pathway, and L-kynurenine from L-tryptophan. The molecular functions for IDO1 are electron carrier activity, indoleamine 2,3-dioxygenase activity, tryptophan 2,3-dioxygenase activity, heme binding, and metal ion binding. The biological processes for IDO1 are cellular nitrogen compound metabolic process, female pregnancy, multicellular organismal response to stress, negative regulation of T-cell apoptotic process, positive regulation of chronic inflammatory response, positive regulation of T-cell apoptotic process, positive regulation of type 2 immune response, small molecule metabolic process, tryptophan catabolic process, cytokine production involved in inflammatory response, kynureninic acid biosynthetic process, negative regulation of interleukin-10 production, negative regulation of T-cell proliferation, positive regulation of interleukin-12 production, positive regulation of T-cell tolerance induction, response to lipopolysaccharide, swimming behavior, and tryptophan catabolic process to kynurenine. When T cells are deprived of tryptophan, they arrest at a mid-G1 phase of the cell cycle. Restoring tryptophan does not restore the activation process, which requires a second round of T cell receptor signaling along with tryptophan. Together, this information has suggested that antigen-presenting cells can employ IDO1 to restrict T cell activation by blocking T cell proliferation due to tryptophan catabolism. A wide range of human cancers such as prostatic, colorectal, pancreatic, cervical, gastric, ovarian, head, lung, etc. overexpress human IDO1. If you have any concerns about the IDO1 gene, contact a practitioner familiar with epigenetics